Hello, my name is Casey. I'm a medical student here at the University of South Dakota Sanford School of Medicine. Today, I'd like to talk to you about colorectal cancer and a compound we've been looking at, UBXN2A, which is a ubiquitin-like protein that we found to alter proteins in the mTORC2 pathway. So the reason why we're studying this is that colorectal cancer is still a leading cause of death. So new pathways or new strategies of combating this need to be looked into, and so that's what we're doing. So first, a little bit of background. So the mTORC2 pathway is a pathway that has been found to be specifically involved in the cell survival and metastasis of colorectal cancer cells, which is why we're targeting it. There are a couple of proteins in the complex itself, the most important being Richter, and there's also downstream proteins such as SGK1 that will be important for this discussion. So we hypothesized then that this UBXN2A compound, which is a tumor suppressor protein, can be upregulated to downregulate proteins in the mTORC2 pathway, thus decreasing the cell survival and metastasis of colorectal cancer cells. So that's what we did. We used Western blot protein stability and flow cytometry and cell migration assays to determine what our results are, and I'll get into that now. So our results have shown we found a compound, veratridine, which can upregulate the tumor suppressor UBXN2A. And we found that that can decrease Richter, the mTORC2 complex protein, as well as its downstream substrate SGK1. The Richter targeting was specific, meaning that UBXN2A upregulation only targeted Richter and no other components of the mTORC2 pathway, which was quite interesting. And we're looking at the middle upper graph here. So you can look at that yourselves. And we've also found that VTD or veratridine can decrease colon cancer migration, which is now at the middle lower left. As you can see in the red bar, the veratridine treated cells have less of a cell migration, indicated as cell index. It's just a measure of cell migration. And cell migration is an important component of tumor metastasis in particular. So decreasing that is promising. The lower right middle graph shows that veratridine administration can simply downregulate stem cell populations, which means that there's less undifferentiated cells and less potential for metastatic stuff. And then finally, in the upper right-hand corner, this is just supporting evidence showing that the UBXN2A promotes the degradation of Richter in a proteasomal degradation fashion. That was doxycycline, which induced UBXN2A and not veratridine in that one. So we can conclude then that UBXN2A can in fact suppress the mTORC2 pathway via decreasing downregulation of Richter, and that veratridine can be the catalyst for this. Some of the limits of the study is this is an in vitro analysis. We simply just have to go in vivo and then clinical results from there. So thank you very much.